Okay, so we're going to get underway. Just a reminder that there is a, on the program that you have, at the bottom there is the Wi-Fi access, which you don't need during these presentations, of course. And as well as at the very bottom, there's a URL for the evaluation at the end. All right, so now we have, every year we always make sure that the students or trainees take part and are very engaged actually here. Many submitted abstracts for consideration for 3 oral talks. So we do have 3 oral talks from PhD candidates, and the first one is by Ahmed Arbain from Dana Devine's lab.
Hello everyone. I will present for you part of my PhD thesis today, which is about the hemostatic function of stored Bethune platelet concentrate in plasma treated with a pathogen inactivation system. Pathogen inactivation system have been introduced to blood bank to decrease transfusion transmitted infection. And this is based on illumination using ultraviolet light with or without photosensitizer targeting the DNA or RNA of the pathogen to inhibit their proliferation. So this technology, the pathogen inactivation system, has led to accelerated development of platelet storage lesion, and it includes increasing in the metabolic activity of the treated units, microphysical release, mitochondrial activity, and also increasing the apoptosis development. The current in vitro quality tests provide no information about the hemostatic function of this unit if it has been transferred to the patients. We were concerned about the hemostatic functionality of this unit after the treatment, since the treated units by this technology has increasing in the safety for the purpose of the transfusion. So we just were worried about the hemostatic functionality of this unit after the transfusion. One of the technology, it's called thromboelastography, and the other one, thromboelastometry, has been developed in Dr. Devine lab to assess the hemostatic state of Bethune platelet concentrate by adapting this technology instead of working with whole blood to be able to work with Bethune platelet concentrate. And unlike other in vitro tests, this technology offers the opportunity to assess the hemostatic state of the blood component in a full coagulation reaction that can simulate the bloodstream under low shear condition. And it's mainly what we are concerned in these parameters is about having the specific parameters which reflect the clot formation time, and it's the time for the clot when it reaches from 2 to 20 millimeters, and also the alpha, which is the rate of the fibrin platelet interaction. We were also interested in the maximum clot formation, which is the maximum firmness that can be reached for this clotting, and the maximum lysis or fibrinolysis for this clot. So the objective was determination of the impact of pathogen inactivation system on the viscoelasticity of platelet derived from Bethune platelet concentrate using the rotational thromboelastometry. In the design and methods, pairs of Bethune platelet concentrate were pooled, and they were splitted to specialized units. And one of the arm, it was treated by the Mirasol treatment, having in that the riboflavin, which is the B2 solutions. And in the sampling, we did sample the units in day 2, day 5, which is the, in Canadian blood surfaces, the units is saved in the shaker under standard condition up to 5 days. But we were interested to see the, how does these units behave after the storage time, which is day 2, from day 2 till day 9. And when we did the sampling, the platelet count was adjusted to 100 by 10 power of 9 cells per liter with using the fresh frozen plasma. Choline and tissue plasminogen activator were used to activate the platelet and to standardize the coagulation level, and also to standardize and challenge the fibrinolysis system in the test. In parallel to that, we used B-selectin to assess the activation level of these units as a quality control test, and also we checked the pH level of the sample when we did the testing. In the results, I will start showing you the in vitro quality test, which reflect the deleterious impact of this illumination on the platelet, where we can see decrease in the pH level in both of the groups. However, the red line represents the illuminated units, where we can see that the pH level decreased under 6.8. 6.8, it represents the lower level of the acceptable units to be transfused. When we look in the activation level, we can see the platelet that has positive for B-selectin marker. We can see significant increase during the storage time for the control unit and also for the illuminated units. However, there was no significant difference between the groups during the storage time for the activation level. It was increased in both of the groups. And now I present for you the results of the thromboelastometry. We can see that there is no significant difference in the clot formation time between the groups in day 2, 5, 7, and 9. However, looking at the maximum clot formation, we can see there is impact from this illumination on the maximum clot firmness, and this impact, it was consistent during the storage time, as you can see it up to day 9 when we tested it. Looking in the fibrin platelet interaction, we can see a significant difference between the groups, and it was mainly after day 7 where we can see a decrease in the rate of the fibrin platelet interaction in the illuminated unit compared to the control unit. In the maximum lysis, we can see that the resistance of the fibrinolysis, it was lower, and we can see here higher fibrinolysis for the illuminated unit, and it was mainly higher in day 9 in illuminated unit compared to the control unit. So what we have to conclude from that, the Mirasol treatment of Buffy-coat platelet concentrate has some impact on the Buffy-coat platelet functionality, and while shear independent in vitro tests show a higher degree of deterioration of the platelet after the treatment, the routine profile indicates that the Mirasol treated BC stored for 7 days are likely to be efficacious for transfusion. At the end, I'd like to thank the volunteers who donated this blood, and to thank everyone who contributed in this study. Thank you for your listening.
So if there's a question, we have time for a question? Bill?
Yes, please.
What?
You just bellow
No,
at you.
no, go. Don't let him bellow.
So if I understood correctly, you did a modified INTEM in the ROTEM protocols. Did you do a FIBTEM, which essentially isolates the contribution of the coagulation factors by inactivating the platelets with cytocolase and D?
Yes, I was working in ROTEM, and I was working also in thromboelastography, which is TIG. So we did kind of that, which is in TIG I used cytokalasin to inhibit the platelet contribution, and in that case I can just see the contribution of the coagulation factors. But in these illuminated units I didn't do it, but maybe it's good.
So you noted a significant decrease in the MCF parameter. One explanation for that would be some kind of inactivation of fibrinogen. Did your other results, were they concordant with that idea?
No, but like I have the other results, which is the activation level using the CD62. But also we are interested to see the fibrin level after the activation of these platelet. One of the possible way is to see the scanning EM to see the difference between the illuminated unit and the control.
Thank you.
Thank you.
Thank you very much.
Thank you.
Our next speaker is Roland Carter from Ed Prysdale's lab.
Hello. All right, so I am Rolanda Carter, and I'm a graduate student in the Prysdale lab. So stroke, heart attacks, and deep vein thrombosis, which are often due to an aberrant clot, are a leading cause of death worldwide. And to prevent and treat these clots, we do use anticoagulants. And since its first approval by the FDA in 1954, warfarin really has been our drug of choice to treat these clots. Because of its nature as a vitamin K antagonist, however, warfarin has multiple different targets within the clotting cascade, which leads to many different disadvantages. It isn't until recently that we were able to have a new option, and this is with the discovery of direct oral anticoagulants, which only affect one clotting factor within the cascade. So today I'll be talking to you about rivaroxaban, which you may have seen on TV as Xarelto, and it was first approved in 2011. And rivaroxaban binds directly to factor Xa to prevent clot formation. I also did studies with apixaban, but I'll leave that for my poster if you come back. Poster number 7. So the reason I got interested in rivaroxaban is because it does bind to one of my favorite proteins, which is factor Xa. And we've discovered a new role for factor Xa in dissolving a blood clot. So when factor Xa, seen here in its intact form as factor Xa alpha, is cleaved in the presence of anionic phospholipid by plasmin, it generates factor Xa beta. And factor Xa beta has this C-terminal lysine that really serves as a binding site for plasminogen, which allows TPA to convert plasminogen into plasmin, and plasmin, as you may know, is the clot-busting enzyme. Further cleavages of factor Xa beta result in a species known as factor Xa 3313, which also has C-terminal lysine, so can generate plasmin. However, Xa 3313 is rapidly turned off in plasma. And what we found is that by covalently modifying the active site of factor Xa or blocking its active site, we were able to prevent the generation of 3313 and thus restore the clot dissolving function of factor Xa beta. And so the question was, can rivaroxaban do this same thing? And this is because rivaroxaban also binds to the active site of factor Xa beta in a similar manner. And I want to tell you that it does. So what we're seeing here is a turbidity assay in which I'm looking at the extent of a clot being formed with an increase in optical density, and then the clot being dissolved with a decrease in optical density. And compared to TPA, which was my control, we see that rivaroxaban rapidly sped up how quickly the clot was dissolved. So to do this experiment, I had to use an excess amount of thrombin in order to bypass the anticoagulant effect of rivaroxaban and thus activating TEN in situ. So what I did is I wanted to see the dependence of factor 10 using 10 deficient plasma. And here what I'm showing you is a graph where I looked at plasmin generation during the course of the experiment. So here we can see that in normal plasma, rivaroxaban enhanced plasmin generation plus 50%. When I removed 10, so using 10 deficient plasma, this was rapidly decreased. And in order to restore an enhancement again, I added back factor 10, suggesting that rivaroxaban enhanced plasmin generation and thus plasma clot dissolution in a 10-dependent manner. Looking at thrombin, we know that when we add rivaroxaban, we might be affecting the in situ generation of thrombin, which could have effect on fibrin structure as well as on the activation of clot dissolving enzymes or inhibitors. such as taffy, which cleaves off these C-terminal lysines that we like. So I did thrombin generation assays where I looked at the amount of thrombin in the absence or presence of rivaroxaban and saw that it was the same. I also look at clot structure and measuring the fiber diameter. There seems to be no significant difference between the two. I also look at the fold enhancement by rivaroxaban and the absence and presence of a TAFI inhibitor, and the fold enhancement seems to be comparable, suggesting that coagulation was well controlled. Now, in all of the experiments that I've shown you so far, rivaroxaban was present while the clot was being formed. So what I wanted to do is remove rivaroxaban from the mix and see what happens. So in this case, I preformed the clot similar to when a patient has a pre-existing clot, and then I added rivaroxaban on top, and then I also add a thrombin inhibitor to inhibit any excess thrombin being present. And what we can see is that compared to TPA alone, rivaroxaban sped up how quickly the plasma clot was dissolved, suggesting a role in thrombolysis for rivaroxaban. Knowing all of this, I then looked into the fragmentation of factor Xa in the presence or absence of rivaroxaban. So here is normal plasma, and we see that we have Xa, which gets rapidly turned into Xa-3313. And just as a reminder, it is Xa-3313 that is rapidly turned off in plasma. And so this is the one that we want to get rid of. And we see that by adding rivaroxaban into the mix, 3313 generation was prevented. And then I had the opportunity to look into patients. And so here is patients that have irregular heartbeats, and they're being treated for rivaroxaban to prevent stroke. And these patients, we see that 10A3313 generation was prevented in all cases. Looking a bit further, I also had patients that were being treated with rivaroxaban for post-surgery, as well as deep vein thrombosis and pulmonary embolism. And we do see that there is a prevention and generation of 33 in most cases. There were 4 patients that had 33 being generated. However, this can be explained by the reduced amount of rivaroxaban that was detected within their plasma. And so this could suggest that rivaroxaban is able to work effectively in these patients because it has a role in dissolving blood clots. Just looking at plasminogen binding, here is a Western blot, and then here is a plasminogen blot. And we do see, I know there's a lot of bands, but just focus on 3313. And we see that 3313 binds plasminogen the most in the absence of rivaroxaban, as expected, but it is rapidly turned off in plasma, whereas in the presence of rivaroxaban, it is factor Xa beta that binds plasminogen the most. So in conclusion, rivaroxaban has a side effect that enhances factor Xa-dependent clot dissolution. So what? Why do we care? Well, 20 to 60% of patients that have an abnormal clot in their deep veins can develop post-thrombotic syndrome. And this is a very painful condition and a chronic condition that is seen mostly as a consequence of ineffective clot dissolution. So by giving these patients rivaroxaban, it's possible that their outcome might be affected. As well, this might hint towards potential applications as rivaroxaban as a thrombolytic adjunct. With that, I would like to thank my supervisor, my lab members, my supervisory committee, my collaborators, and funding. And I will be happy to take any questions and also encourage you to come by poster number 7 for further discussions.
You mentioned about post-thrombotic syndrome. So have you looked at patients who have post-thrombotic syndrome and seeing versus those who don't get it in terms of expression of?
So I haven't looked at any of the patients that had post-thrombotic syndrome. I do know that there is a study where they looked at how patients do that are being treated with rivaroxaban, and those patients had less recurrent venous thromboembolism and had a successful outcome compared to those that didn't get treated with rivaroxaban.
And are you developing an assay to look at that fragment?
It is something that I guess we could consider. Yeah, we see the fragment, then it's probably the case.
Okay. Anybody else? Oh, there was a question. We need a mic. Oh, we got a mic.
Most people who take rivaroxaban have atrial fibrillation. They are old and they often fall, and when they hit their heads, they bleed. There isn't now an antidote for rivaroxaban at an Exodex, but it has a very short half-life, and you show that this effect is in fact quite long. Is that going to be a problem?
It's possible. Yeah.
That's a good answer.
Yeah.
Okay, I got one more question at the back.
You looked at a bunch of different clinical conditions in patients who take rivaroxaban. One clinical condition that people often take rivaroxaban for a clot that they developed and end up being diagnosed with malignancy afterwards. And so rivaroxaban's not approved for malignancy-associated clots, but a lot of people end up on it. I wonder if you looked at that population at all, and maybe even to see if this protein fragment is present, or if higher doses might work or whatnot. I think that's one area of the novel oral anticoagulants that hasn't been well sorted out.
Yeah, so this was a really kind of exploratory study to see exactly what patients or what the fragments look like in patients. So we knew what it looked like when we took normal plasma and we spiked it with rivaroxaban. And so here we only had the opportunity so far to look at those three groups and haven't gone beyond that. But it's possible that if those patients are being treated with rivaroxaban, that we would see that kind of a factor 10 band present in their plasma.
Okay. They can find you at poster what?
Number 7.
Okay, thanks. And our third speaker, PhD candidate speaker, is Vivian Chan from the lab of Dr. Christian Castro.
Good morning, everyone. Thank you for allowing me the opportunity to speak to you this morning. So today I'll be telling you about my project as part of my PhD thesis, which is the controlled transcription of exogenous RNA in human platelets. So being in the Center for Blood Research, or as blood researchers in general, I'm sure all of you are very familiar with platelets' role as hemostatic, as mediating the hemostatic response. But let me shift your focus a little bit this morning and think of platelets instead as delivery vehicles, because they really are the ultimate drug delivery systems that exist in our bodies. So platelets mainly circulate in our bodies in a resting state while containing many procoagulant molecules, which would be a disaster if they were just released everywhere. But what platelets are very good at doing is that they can seek out areas of vascular damage, and at these wound sites, they are able to activate, aggregate, and most importantly, release very bioactive molecules, which includes small molecules, proteins, and nucleic acids. So while platelets have the potential to be very good drug delivery systems, the challenge really exists in loading platelets with exogenous therapeutics. This is partly because platelets do not have nuclei of their own, so traditional genetic manipulations techniques that are available are not really applicable to platelets. So in work that we've been doing for the past few years and that we recently published, we hypothesized that we can transfect platelets with synthetic nuclei. So if we're able to give platelets these functional units containing all the machinery necessary for transcription, we hypothesize that we can then upregulate or downregulate certain protein targets inside of platelets. What we did was we developed these nanoliposomes, which consisted of a lipid bilayer encapsulating an aqueous core. And inside of these nanoliposomes, we encapsulated a DNA template, nucleotides, as well as a T7 RNA polymerase. We photocaged one of the nucleotides in order to suppress the transcription of RNA until light is shined on the samples and we get RNA being made inside of the liposomes. So we thought that we could give these liposomes to the platelets, get the platelets to take up the liposomes and be able to transcribe RNA inside of the platelets, allowing us to really regulate gene expression in platelets. So next I'll just give you very briefly some of the data we have on this. So first, we determined that we can controllably initiate RNA transcription using the photocage nucleotides. So we're able to suppress the transcription and get the transcription to be reactivated upon light stimulation. We were able to do this both in solution here and also in liposomes, where we could transcribe a GFP mRNA, a luciferase mRNA, and if we took the luciferase mRNA and translated that into protein, we found that it made functional luciferase, suggesting to us that we were making full-length RNA. We then took the liposomes and included a fluorescent lipid into the bilayer and gave them to the platelets, and found that platelets incubated with these liposomes had an increased liposome fluorescence compared to untreated platelets. And using confocal microscopy, we confirmed that the platelets were indeed taking up the nanoliposomes. We also used several chemical inhibitors, such as sodium azide and cytoclassin D, which are just general metabolic inhibitors, as well as a specific inhibitor, dinosaur, for clathrin and caveolate-mediated endocytosis. And using these inhibitors, we were able to confirm that platelets were endocytosing these liposomes that we were giving them. So using the functional liposomes, we found that platelets incubated with these liposomes and incubated for an hour could produce RNA, whereas platelets that were first lysed or inhibited with the small molecules did not show an increase in mRNA after an hour. So this is, to our knowledge, the first case of a exogenous reporter RNA being transcribed inside of platelets, and really opens the doors to many applications to be used with the platelets. So what I've shown you today so far is that we can load platelets with these transcriptionally functional liposomes. And we also have preliminary unpublished data showing that we can deliver just purified RNA to the platelets in greater efficiency. And some of the applications we're thinking of is that if we deliver these RNA to the platelets, can we induce a RNAi response or get the mRNA to be translated in the platelets? And these really will allow us to upregulate or downregulate certain protein targets and potentially be able to study platelet function in ways that we haven't been able to before. What's also exciting is that we can activate these platelets and look for the release of the exogenous biomolecules that we give them, and whether these can have an effect at the site of activation. So if you guys have any cool applications or ideas, I invite you to come to poster 8 after you talk to Rolinda at poster 7. And with that, I'll just thank the members of the Castrop Lab and Stephanie Nowakowski in particular, as well as our collaborators, my supervisory committee, the funding sources, and CBR for this opportunity to talk to you guys. Thank you.
So question, so I have an application. So it sounds like an improved approach to treat balding. Isn't that true? Right, thank you. Any other serious questions? Or not so serious? No? Bill?
Could you test the sensitivity of the system by putting in DNA for T7 RNA polymerase instead of putting in the enzyme?
So putting a template for the T7 polymerase itself. There would still need to be a source for some kind of RNA polymerase to transcribe the DNA, because platelets will not have the polymerase
There's nothing
itself.
endogenous.
Yeah.
Okay. Thank you.
That was a really nice talk, Vivian. How efficient was your uptake of the vector?
Not very efficient, to be honest.
And are you looking into ways to improving that? Do you have secrets going on?
Yeah, not quite secrets, but so there are many ways to increase uptake of lipid particles into cells in general. So we're looking into some clinically approved formulations of these lipids to increase the efficiency, but how well they actually work in platelets is still to be determined.
That sounds pretty secret.
Okay, thanks very much.